Hello all, I am Reddy Rani, a PhD student at International Institute of Information Technology, Bangalore, India. In this talk, I am going to present our work titled Construction and Visualization of Diseasome of Lung Diseases Associated with COVID-19 from Co-Association Networks of Multi-omics Data. Our motivation in this work is to construct a diseasome, that is, a bipartite graph with comorbidities associated with COVID-19 and genes identified using multi-layer correlation networks. Diseasome is a bipartite graph consisting of two disjoint sets of vertices with only interset edges mapping between the disjoint sets. We propose to use diseases that are associated with COVID-19 such as tuberculosis, lung squamous cell carcinoma and its subtypes, pneumonia and etc. for the diseases set, and the genes associated with these diseases as the second set for the bipartite graph. We demonstrate our workflow with the multi-omics values such as DNA methylation features and mRNA expression traits of lung carcinoma obtained from the Cancer Genome Atlas. A bipartite graph of the diseasome is highly expected to be unbalanced. That is, disease genome set in the bipartite network can be far greater than disease phenome set. We propose to visualize diseasome using our novel graph layout called Ratrix. Ratrix is a composite of two visualizations in a single view, that is, matrix visualization for diseases set and circular layout for the genes set of the bipartite graph. This layout uses quadratic spatial complexity of matrix and circular view. The nodes on the circumference of the circle in the layout are placed using polar coordinates. The example Ratrix layout shown here is with 5 diseases in matrix view, 73 genes on circular view, and 209 edges connecting both. Both genes and diseases are seriated based on their node degrees and set cardinalities respectively. In our work, a multi-layer network is defined as network with separate layers with no interlayer edges and with the possibility of non-empty intersection set of vertices across the layers. We measure partial correlation coefficients between the mRNAs for layer 1 and Spearman correlation coefficients between methylation features and mRNAs for layer 2. Partial correlations is computed as a linear correlation between residual errors of the regression model. We use Lasso model for multivariate multiple regression computation. Spearman correlation is a distribution-free statistical procedure, and unlike Pearson's correlation, this method is not a linear correlation measure. For the case study to create multi-layer networks and identify disease-specific genes, we have used lung carcinoma dataset published by Wang and team. The data is with 106 samples, 23,704 methylation features, and 12,042 mRNA expression traits. We mapped the DNA methylation probe IDs to gene symbols using our bioconductor Illumina package. Both the omic feature values are standardized to have zero mean and unit variance. Genes that are neither significant and nor relevant are filtered using threshold values. We have used percolation analysis to find edge cutoff value. When a graph is explored with sequential steps of threshold values, it can be seen that at a particular threshold value, the joint connected component will start dividing into multiple subcomponents. Percolation analysis works on the same principle. The identified threshold values using percolation analysis for layer 1 and layer 2 are 0.38 and 0.44 respectively. Genes in a community are expected to have high similar functions and are also highly correlated among themselves. We have implemented a generalized low-vein community detection method on layer 1 and biclustering on layer 2. Layer 2 is with 2 genomic features And biclustering is used as it can group methylation features and expression traits simultaneously. In our work, we found 8 communities in layer 1 and 11 communities in layer 2. We ranked the genes of each cluster using edge weight density value. Edge weight density of a gene is the ratio of the sum of edge weights of edges containing that gene to the total number of non-zero edges. We use the union of the genes in communities of both layers to validate. The top 10% of the ranked nodes are used to verify functional annotation and biological significance using a gene annotation tool, David. As shown in this table, our method is successful in identifying significant genes of lung carcinoma. We have proposed a workflow to identify significant genes using multi-omics data. We have used lung carcinoma data as a case study and demonstrated how our workflow is successful in identifying significant genes. We have introduced our novel graph layout matrix to visualize the diseasome. We intend to implement our workflow with the comorbidities associated with COVID-19 to create a diseasome between lung diseases and relevant genes. Thank you for your attention.